We've looked at the chambers of the heart, at the valves, at some of the major vessels of the heart. Now it's time to zoom in and start looking at heart muscle itself. So let's look at some microscopic anatomy of cardiac muscle. So cardiac muscle cells are striated, short, fat, branched, and interconnected, which coincidentally is an exact description of my girlfriend my freshman year of high school. Let's take a look at what that means. Striated, just like skeletal muscle, remember? It has the A band and the H zone and the I band and so on. That's all because of the same things. Short, so these muscle fibers are shorter than skeletal muscle and they're thicker. They're also branched and interconnected. So look in the bottom illustration. You can see that the heart muscle fibers, myocardium fibers, are connected to one another and they branch. Why do you suppose they do that? Well, the reason is that you never want one part of the heart to be beating and another part of the heart not to be beating. The heart has to beat as a unit, otherwise it will not work. So all these muscle fibers are therefore connected together and they branch together so that the heart beats as one giant single unit. All right? There is a connective tissue matrix, the endomysium, connected to the fibrous skeleton. Remember we saw the fibrous skeleton before. That's a fibrous tissue band that basically holds all the myocardium in place. T-tubules are wider but less numerous. Remember, T-tubules are how the action potential, Remember the muscle action potential got down inside of the sarcomere that way. Sarcoplasmic reticulum is simpler than the skeletal muscle. And there are numerous large mitochondria, 25 to 35% of cell volume. So why do we have so many more mitochondria? I mean, think of most of the skeletal muscles in your body. They're only moving now and then. Your heart muscle doesn't ever stop until you die. So the heart muscle needs a ton of ATP. Therefore, you have lots of mitochondria where you make the ATP. So here we can see the branched interconnected fibers up in the upper right illustration. There are what are called intercalated discs. Intercalated discs are junctions between these cardiac cells. There you can see in the lower part of the upper part of the illustration on the right, you can see the intercalated disc joining the two myocytes together. There are desmosomes to prevent cells from separating during contraction. Remember, they called those in Bio 156 spot welds, maybe. They hold cells together. And then there are gap junctions. Gap junctions allow ions to pass, and therefore you can electrically couple adjacent cells. So again, in order to make sure the heart beats as a single unit, ions can flow freely from one cell to another through the gap junctions. They say that the heart muscle behaves as a functional syncytium. A syncytium is an old-fashioned word, but it's a cool word. It basically means a bunch of parts that all work together as one whole, as one entire thing. And that's the way the heart has to work. If one side of the heart is pumping and the other side of the heart is not pumping, that doesn't work. The entire heart has to contract and pump as a single unit. So how is myocardium different from skeletal muscle? Do you remember what we just talked about? Myocardium does not have T-tubules. Only skeletal muscle has sarcoplasmic reticulum. Only skeletal muscle is multinucleated. Myocardium does not have mitochondria. Only myocardium has intercalated discs. Which one of those is the defining feature? Yeah, that's how they're different. All the others are the same. It has T-tubules. There is sarcoplasmic reticulum, SR. They are multinucleated fibers. They have lots of mitochondria, but only heart muscle has intercalated discs. That's because only your heart muscle is branched and interconnected, okay? Let's look at how the heart muscle contracts. Depolarization of the heart is rhythmic and spontaneous, which coincidentally describes my girlfriend my sophomore year of high school. She was a riot. Had some crazy times with her. So what's going to happen here is that about 1% of cardiac cells are autorhythmic, self-excitable. Self-excitable is my girlfriend my third year of high school. So what does this mean? Most of the cells in your heart are myocardium. They're heart muscle cells. But the heart has its own little electrical system. Those are the autorhythmic cells. They are self-excitable, meaning that they don't need to be stimulated by a nerve. they create their own pulses, their own electrical impulses, all right? Gap junctions ensure, again, that the heart contracts as a unit, and the heart has a long absolute refractory period. So what happens here is once the heart beats, think of the nervous system. Once a neuron fires an action potential, it could fire another one in about 1 millisecond later. It had a refractory period of about 1 millisecond. The heart muscle has an absolute refractory period of 250. Why? Why would the heart muscle have such a longer refractory period? Well, remember, what's the heart's job? The heart's job is to pump blood. Is there always blood everywhere in the heart? No, blood has to come back from the lungs or from the body. Remember, it trickles into the atria and then very down eventually into the ventricles. Well, it takes time for the heart to fill with blood. That's why the heart's own internal nervous system has a very long refractory period. Comparatively, 250 milliseconds is 1/4 of a second. So you need 1/4 of a second for the heart to fill with blood again. And there you can see in the upper right diagram, you can see the sinoatrial node, says it's the pacemaker. So those are the self-excitable cells. Those are the autorhythmic cells. Those are the 1%. They are generating their own electrical impulses. They don't have to get a signal from the brain. They generate their own electrical impulses by themselves. So again, long refractory period, the heart needs time to refill. If your heart is pumping all the time, it's contracting all the time, rather, that's called fibrillation. Fibrillation is very dangerous because if the heart is fibrillating, that means it's pumping before there's any blood in there. It means you're just empty pumping. You're not actually moving any blood. That's a very serious condition. Okay, so continuing here with contraction. Down on the right, you see sodium and calcium. We have action potentials here in the heart, and let's take a look at them. So depolarization opens voltage-gated fast sodium channels in the sarcolemma. So in other words, once the pacemaker fires, all right, so once the heart fires its electrical signal, that causes, we're looking at cardiac muscle contraction now. We're looking at what happens inside of a cardiac muscle cell, not a pacemaker cell, not the autorhythmic cells. We're looking at the actual muscle cells themselves. What happens is the depolarization that comes from the pacemaker in the heart muscle cell itself, that opens voltage-gated fast sodium channels. Remember, that was like the action potential that we saw in the nervous system. The first thing that opens are sodium channels, all right? Those depolarize the heart muscle, all right? The depolarization opens those channels. We have a reversal of membrane potential from -90 to +30. So remember in the nervous system when we studied it was -70 was the resting membrane potential. In the heart it's more like -90. But just like in the nervous system, we depolarize up to like +30. So there's a massive reversal of membrane potential from -90 all the way up to +30. Depolarization wave in the T tubules causes the sarcoplasmic reticulum to release calcium. So just like in skeletal muscle, as the action potential travels down the T tubules into the sarcoplasmic reticulum, it causes it to release calcium. All right. The EC coupling occurs as calcium binds to troponin and the binding of the filaments, sliding of the filaments rather, begins. This is just like skeletal muscle. You should go back and review your skeletal muscle slides because it's all basically the same thing. Remember, troponin, tropomyosin complex blocked the myosin binding sites on the actin filaments. Remember that the myosin head had to grab a hold so that you can row the filaments, do the sliding filament mechanism. So what's happening now is that just like in skeletal muscle, once the heart's pacemaker fires, that causes, the depolarization causes the voltage-gated fast sodium channels to open. They depolarize the membrane. That depolarization wave goes down the T tubules, causes the opening of calcium, and calcium works just the same way in heart as it did in skeletal muscle. Okay, continuing. The depolarization wave also opens slow calcium channels in the sarcolemma. So the heart muscle cells have these slow calcium channels. The sodium channels we said were fast sodium channels. They open and shut really quickly, just like in skeletal muscle. There are also slow calcium channels. They open slowly and they take a while to close again. Why does that happen? That prolongs the release of calcium. So look in the bottom half of the illustration. See the green graph? So notice where it says 1. 1 would be where the pacemaker fired the action potential. 2 is the opening of these fast sodium channels. 3 is where they peak, they top out, they close now. Now finally the slow calcium channels are opening, and that's what's happening at 4. Remember, we didn't have these calcium channels in the nerve action potential, okay? Those weren't not there. This is unique to heart. So those slow calcium channels open, that causes the prolongation of the action potential. What that means is it makes the heart muscle contract longer. Why would we want the muscle to contract for a longer period of time? Because we've got to pump all the blood out. There's no good of having a quick contraction of heart muscle. That would only pump a little bit of blood. You want the muscle to contract for enough time to really squeeze all the blood out. So that's what these slow calcium channels are for. They allow for the heart muscle to continue to squeeze blood out. All right? Duration of the action potential in the contractile phase is much greater in cardiac muscle than in skeletal muscle, and that's in order to squeeze out as much blood as possible. So notice then, reviewing from the start, we had down at the green graph, the pacemaker fires at 1, sodium channels open at 2, that depolarizes the membrane, allows the calcium to be released, to cause the troponin and tropomyosin to move away from the actin, so the myosin can grab a hold. Now that the myosin has grabbed a hold, we open those slow calcium channels so that we contract the heart for an extended period of time so that they will keep squeezing that blood out. They will keep sliding those filaments. They will keep contracting and continue to pump that blood out. Then repolarization, once the calcium channels have closed, the slow calcium channels, then we have potassium channels that open, just like we had in the action potential. So these potassium channels open, and they hyperpolarize and repolarize. They bring the resting potential back to normal again. All right. So in a way, the cardiac muscle action potential is very similar to the normal nerve action potential, but we have the extra part of the calcium channels, and those are to make sure that we stay contracted to pump all that blood out. And there you see it right there. I've been known to put this graph on quizzes and exams and ask you to explain it, all right? So once again, on the left side, once the pacemaker fires in the heart, the self-excitable, the autorhythmic cells, then we have the fast sodium channels, those depolarize the membrane. Then we open the slow calcium channels, that maintains contraction to pump all the blood out. Those close, and then we open the voltage-gated potassium channels, and those restore. As potassium leaks out, that restores the membrane potential back down to around -90, okay? So the electrical events in the heart has, as we've talked about, has its own built-in electrical system. Let's take a look. So it has an intrinsic contraction system, or a network of non-contractile autorhythmic cells that initiate and distribute impulses to coordinate the depolarization and contraction of the heart. So we're done now looking at the heart muscle cells. We're now going to look at those autorhythmic cells, at those pacemakers. And we're going to see that there are actually two pacemakers in the heart, the sinoatrial node, which I mentioned, and now we can also see there's an atrioventricular node. So these are the cells that are creating the electrical events in the heart. These are its own intrinsic, its own built-in electrical system. The autorhythmic cells, they're not contractile. These are not making the heart muscle contract. These are simply sending action potentials through the heart muscle, okay? Neurons have a refractory period of about 1 millisecond, but cardiac muscle has a refractory period of about 250 milliseconds. Why is that? Is it to allow cardiac muscle time to rest because it's tired? To allow ion concentrations time to return to equilibrium before the next contraction? To make sure that cardiac muscle is not damaged by contractions that are too close together, that poor heart? To allow the heart time to refill before the next contraction? Or to make sure that all the blood is pumped out? Remember, I'm asking about the refractory period. Yeah, that's to allow the heart time to refill. All right. So let's look at these autorhythmic cells. Let's look at how they work. We saw how the cardiac muscle cells, the contractile cells, how those work. Now let's look at how the autorhythmic cells work. So now we have another action potential here. Heart has two different kinds of action potentials. One for the contractile myocardial cells, and another type of action potential for the autorhythmic cells. We already looked at the heart muscle action potential. Now let's look at the pacemaker, the autorhythmic cells. Let's look at their action potential. So in this case, we have what are called slow sodium channels. And here's the deal. These are sodium channels that are leaky, all right? They kind of like are always allowing sodium to leak in. So remember, in the skeletal muscle, we had graded potentials that, summed in the axon hillock, and they brought us from the resting membrane potential up to threshold. Well, it's the same thing here. The autorhythmic cells are normally down at around -60. Well, these slow, leaky sodium channels are slowly letting sodium leak in until after a period of time, we finally make it up to the threshold for these autorhythmic cells, which is -40. Instead of -55, like in the nervous system, it's now -40. But that's what's going on. So in other words, nobody has to tell them when to fire. They're slowly leaking sodium, and eventually they leak enough sodium to reach threshold, and that's when they fire, all right? At threshold, calcium channels open. So now the rising phase of the action potential. So unlike in the nervous system and unlike in the myocardial cells, now the rising phase is calcium. All right. It was sodium in those other cases. It was sodium in the nervous system. It was sodium in the heart cell contractile muscle cells. Now in the autorhythmic cells in the pacemaker, the ascending portion of the action potential is calcium channels. All right. Calcium influx produces the rising phase of the action potential, and then once those close, it's potassium that brings us back to normal. So these fast potassium channels open, and potassium flows out of the membrane. That returns us back down to -60. And then look, the whole thing repeats itself. See? It's kind of like one of those machines that, that automatically resets itself. I remember there were these things like there was a little post and like a little woodpecker thing, and there was a weight on the end, and it would peck, and then it would come back, and then peck and come back. I don't know if you have any idea what the hell I'm talking about, but what's going on here is that the slow sodium channels just sort of guarantee that there will be a delay between each time that the pacemaker fires. See? It takes a while for the sodium to leak in. Once it hits threshold, then kaboom! The calcium channels open, the pacemaker fires, and then the potassium channels open. Now the pacemaker goes back to its resting potential, and now it's going to take a while as the sodium slowly leaks in until we reach threshold again, and then boom, we fire another pacemaker potential. So see, it automatically governs its own time lapse here, all right? See, it automatically controls how often it fires with those slow sodium channels. So here you see a nice graph that puts the whole thing into one picture. If I were you, I would sit down and spend a little time with this and just see how that works, okay? These are the autorhythmic, the pacemaker cells. They don't need anybody to tell them when to fire. They, in fact, tell the heart muscle when to fire. So how do they know when to fire? Because they have these leaky sodium channels that take time to let enough to leak in to hit threshold. And when they do hit threshold, that's when they fire.